This is your quick guide to the different levels of protein structure. Okay, so proteins fold themselves into 4 different structure levels, which are each held together by different bonds, and we're going to go through the 4 here. So first up we have our primary structure. And the primary structure is just the different amino acids which are held together by peptide bonds. So these are amino acids. This could be G, this could be C, A, T. And then they're held together by peptide bonds which are formed in a condensation reaction to form a polypeptide. Next up we have the secondary structure. So the secondary structure is the different local shapes that these polypeptide chains form, and these are held together by hydrogen bonds between the different amino acids in the chain. So there's 2 different possibilities. We're either going to get a beta pleated sheet, which we can see here, or we're going to get an alpha helix, like we can see here. And then these hydrogen bonds will form across here between these different amino acids, and they will hold these different shapes in place within the protein. Next up we have the tertiary structure. So tertiary structure is the overall 3D folding of the whole polypeptide chain. And we can see 2 different forms here. We have the fibrous protein or the globular protein. So within these structures we'll still get hydrogen bonds forming, but the main bonds to hold this in place are the bonds which form from the R groups of the different amino acids. And these include ionic bonds between different polar molecules, and also disulfide bridges which form between amino acids that have sulfur in their R group, and these are much stronger, so they help to hold this overall 3D structure in place. You're also going to get hydrophilic groups, and they're going to tend to stick towards the center of the proteins where they can shelter themselves from any water molecules. And then finally we have the quaternary structure. And the quaternary structure has the same bonds as the tertiary structure, but instead of having one polypeptide chain, we now have multiple polypeptide chains joining together to form this protein. So this could be something like hemoglobin, which contains 4 different polypeptide chains joined together. Okay, so remember, primary is your amino acid chain, secondary is the local shape that's formed by hydrogen bonds between the different amino acids, tertiary is the overall 3D shape of the protein. This includes hydrogen bonds, but also has ionic bonding and disulfide bridges, and then the quaternary structure is the same as the tertiary, but now you're adding in additional polypeptide chains. And that's it.